Good morning and welcome to our seminar, Evolving Analytics in the Upstream Lab by Using the Power of LCMS. I'm Laetitia Denby, Biopharmaceutical Regional Marketing Manager at Waters, and I will be your moderator today. In many organizations, there is a growing need to optimize bioprocess development and validation by incorporating elements of the quality by design approach. This should allow to reduce the costs and increase the speed of development. But it will also add flexibility to the process and improve the overall quality of the product. Each phase has specific goals and characteristics which drives the analytical needs. The benefits of LCMS and LCMS assays have become clear for both product critical quality attribute analysis and media monitoring. But up until now, they were mainly performed in dedicated analytical labs far from the process stream, leading in delays in decision. Now, with instruments such as the BioQuad system, advanced analytics can be performed directly at the point of decision, and reliable and reproducible data can be generated even by non-MS expert users. Today, we will present to you two different LCMS workflows that will apply to bioprocess development. First, Mark Rona, Principal Scientist at Waters, will show you how BioQuad can be used to monitor nutrients in spent cell culture media. And then, Charles Prochaska, R&D engineer at Sartorius, will show you how the same BioQuad system, in conjunction with the Umber system, can allow rapid turnaround times of map product quality data on critical quality attributes. And now it's up to you, Mark.
Thanks, Laetitia. My name is Mark Rona, and I'm a Principal Application Scientist here at Waters. I'm excited to share some data with you today on nutrient monitoring in spent cell culture media, which we've been doing in-house and with a few collaborators. Before I begin my presentation, I would like to acknowledge my team, and especially Yun Ali Yunus as the primary contributor of this work. I'd like also to acknowledge Elsa Gore and Andy Mahan from Johnson & Johnson for their many insights and help with cell culture media analysis. Let's start with what cell culture media analysis is and what our customers and collaborators have been asking us in terms of solutions to help them facilitate it. We know that the primary function of media is to maintain the health, good growth characteristics of our clones in order to produce the product we want at high quality and with high yield or titer. As the media is consumed, we want to measure and track the primary components that are consumed to produce the therapeutic itself. As cell growth increases, consumption increases, product is produced, and metabolites are generated in response. These changes occur for a large number of reasons and may be products of energy pathways, markers of cell growth, viability, death, and other signals over time. The more we monitor and track, the more information we can have to determine their relationship to measurements such as titer, viability, and product quality. As the process is scaled up or gets closer to QC or manufacturing, analytics employed need to be increasingly simple and easy to deploy. We need to make sure that the analytics we collect are available to make decisions and provide insight across a large part of the process. So where are we today? These are some of the techniques we see our collaborators use to measure both media components as well as metabolic byproducts and fundamental chemical properties. Many of the current techniques in use are fast and measure a set number of components and do it well. In general, we see that these technologies are dominated by speed and throughput, but also limited targets that we exclusively know about. Some approaches like the Accutag technology produced by Waters enable simple measurement of large number of components using derivatized approaches. We've also worked with many customers on targeted LCMS approaches in the past, such as the Waters TQS micro system here, where we can start to measure larger numbers of panels. So if we develop tools that measure more components and bring them closer to the process itself, they need to be both information-rich and easy to use. Cell culture media is actually very complex and in flux during the entire process. We know that the current tools only measure the tip of the iceberg. We want to know what's happening in the media in response to specific product quality attributes changing in drug substance. There's a lot more to these samples than just base amino acids and starting feed components. Can we use this information to monitor or inform the process? There are a number of areas we've been working with to analyze and provide insights. For feed and raw materials, we can assess quality and levels of many different components. We can compare feeds from different vendors or batch comparisons and assess the differences for the listed ingredients and impurities. We can look at either serum-based or chemically derived media, which also may be customized with additional trade secret ingredients. For spent media analysis, we can understand the feed characteristics, monitor metabolites of interest, or find new metabolites being excreted into the media. We've had interest from people looking at both clone selection as well as production scale. We can compare media consumption and turnover across clones and batches. Areas of high interest include looking at metabolic profiles and metabolism products that are present when we see product variability such as sequence variants, glycan profile changes, or increased trisulfide bond formation. For process development and scale-up, we can see changes across a wide variety of metabolites. Seeing these changes enables teams to better understand what feed schedule, additives, timings, and scaling factors are critical during this phase of the process. This means that the ideal solution would let us analyze both the things we know about and also observe changes in the media directly attributable to the process itself. So how do we collect the data? We start with an easy-to-use LC-MS system that lets us measure many of the targeted components. This lets us review targets of interest and see how they're changing across clones or batches or time. Once we review these targets, we can dig into the data to find additional metabolites of interest in our data set. What do I mean when I say metabolites of interest? These would be additional compounds that mass spectrometry can observe that track with the changes we see in the various bioreactors. The rich data sets provided by LC-MS can also utilize statistical tools like multivariate analysis, and I'll talk a little bit later about how we can interrogate the data and begin to correlate and more deeply understand these changes and variability found across bioreactors. This is the overall system. We start with a large library which was built using authentic standards. A comprehensive analysis is performed using LC-MS, measuring the components we have in this library, and is performed using our newest premier separation technologies. We chose to build the solution on the most deployable and easy-to-use mass spec system that Waters has ever made, the BioAccord. And finally, using informatics, we can access both the targeted information using the library of knowns, as well as doing further analysis for new metabolites and unexpected changes. The first step in building a comprehensive method is to have a comprehensive library of components. Here is a general overview of the classes of compounds that can be measured today using the platform. The base package currently includes amino acids, their derivatives, organic acids, nucleic acids, and base nucleotides, as well as vitamins and other compounds. It offers a broad coverage for not only targeted work, but also enables us to see new metabolites and dig more deeply into the data for additional changes and components to monitor in the future. In order to keep track of all this information, Waters Connect uses an integrated library. Here we see one of the compounds we routinely measure. The information for the 200 compounds in the library was all generated using authentic standards. With authentic standards, we are able to get precise retention times and confirmation of major fragments that can be used to help routinely monitor the samples. Essentially, we have a fingerprint and essential information recorded for each molecule, which ensures correct identification and specificity at each time we run a sample. The main method is a sensitive and specific reverse phase chromatographic method, which was developed using our latest premier technologies. On the bottom, we can see an overlay of many of the targeted components we measure in cell culture media for a single injection. This is what's going on in every single sample across all the time points in all the reactors. But don't worry about the number of peaks, as we have a capability to resolve these using the BioAccord system. The BioAccord is a high-resolution mass spectrometer. Analytically, it measures the mass to charge of ions. This is a property that's related to the molecular weight of each component, usually by the addition of a single proton of mass. For each component in the spent cell media, we can uniquely identify it by its mass to charge as well as its retention time. Measuring these properties means we can simplify this analysis immensely and review the data logically while continuing to collect all this data for each sample. The benefit of using this technology is that we can target and observe new compounds as well that may be specific to a given process or set of conditions, or even unexpected changes in the bioreactor. Instead of overlaying the data, because we know the mass to charge and retention time of each component, the BioAccord can resolve all of this information into independent chromatograms. Here we can see many of the different compound classes that we can measure with a single method. We can also see that using ultra-performance liquid chromatography and our Premier technology enables great peak shape and separation performance of many compounds that are often quite challenging to separate and analyze confidently. The more samples that we run, the more information we can collect about batches and over time. The method was developed to be fast, accurate, and reproducible. Here we see a snapshot of how some of these components behave analytically. We can see that the standard deviations for the retention time, the mass error, and the response provide a lot of confidence to measurements in these samples. Waters has recently introduced a number of next-generation chemistry and separation products focused on high-performance surfaces, which help deal with challenging analytical problems. The Premier line of column and LC systems products enables the highest performance separations possible. It offers better, more reproducible chromatography, increased recovery and sensitivity by reducing the secondary and undesirable interactions between the analyte and all the surfaces it comes into contact with. These extra interactions are what often yield poor peak shape, poor recovery, or variable results. Here we see a representative trace of a number of compounds that are challenging to separate without derivatization or specialized methods. This technology is perfectly suited for the challenges we face when we run cell culture media. Here's an example of the type of separations we see for some of the challenging isobaric species commonly found in cell culture media. For isocitric and citric acid, as well as isoleucine and leucine, we see baseline separation. This type of specificity and peak shape is critical for measurements of these components repeatedly and across challenging concentrations, often seen in spent media or feed samples. The BioAccord itself has actually already been deployed for a number of areas. It was designed to routinely measure a large number of key biotherapeutic product attributes, including intact and subunit mass analysis, peptide mapping, and released N-glycans. We use the same system for generating this data. It is ready to use with fully automated calibration and was designed to generate high quality and rich information while at the same time being easily deployable and accessible to non-mass spec experts. All of this technology comes in a small footprint and intuitive interface. Later on in this session, you'll hear from Charles Prochaska from Sartorius on some of the other capabilities of the BioAccord. Here's the core workflow that comes with the solution. We start with tools that enable a very high-level review of samples in our run. It's a broad overview of our injected samples. There are many components that can be monitored in cell culture media, so the workflow breaks down the review of components into easy-to-digest classes and steps. The workflow can also be customized to look at specific pathways or combinations of metabolites that may be related to a specific process of interest. There is a step to review calibrations, and once we've reviewed all the components, the data is also organized into summary tables which are exportable to spreadsheets or other analytical packages. Here's an example of looking at a single component and then observing the trends of that component over time. On the left, the software displays the chromatogram of the currently selected component, in this case choline, which is a vitamin of interest. On the main panel, we can see the response of choline across all 12 bioreactors in a clone selection experiment. Each clone shows 6 time points per reactor, which are run in duplicate. This view makes it pretty clear which samples are changing the most for any given analyte measured in the assay, and can give you a good sense of the differences between, say, reactors 1 and 2, or any other bioreactor, and lets you compare them all simultaneously. Beyond the targeted compounds, there is a lot more to cell culture media. Once we finish the review of targeted compounds, we are also able to look for other not yet identified metabolites that are changing across the bioreactors. Here's an example where Yun detected another component in the data that was tracking with changes observed in the clones. On the left, we're showing the spectrum detected for the unknown compound. We have both the intact or precursor mass of the compound and its isotope pattern precisely measured. Directly below, we see the fragmentation spectrum for this compound, which we can also generate on the BioAccord. Once we feed this information into the Discovery Toolkit workflow, the software calculates plausible elemental compositions and fragmentation patterns. We can optionally link up to ChemSpider to search multiple databases for matches. The database hits then can be ranked by looking at the fragmentation patterns. In this case, Yun determined cysteine glutathione disulfide was the most likely candidate molecule based on the information. We then went back and verified this hit by ordering an authentic standard and confirmed the fragmentation pattern and the retention time. We then added the compound to the library and began routinely screening for it. We can repeat this process and customize a method for any new or proprietary compound of interest. In order to find components of interest across many samples, multivariate analysis tools provide a valuable starting point. Waters Connect integrates with multivariate data packages, and here I show some of the data we export to EasyInfo. The color coding represents the days for each clone, and we can get a sense of this change and the differences amongst the clones by doing a principal component analysis. If we consider the clone selection process itself, we would expect that early on in the experiment, the samples would be very similar to one another, and that's exactly what we see. Early on, the data showing here in the bottom right quadrant shows the data for day 0 and day 2 for all of the clones in red and black dots. And so early on in the experiment, you'd expect them to be very similar because the feed is all the same. They've not had a chance to differentiate in the products they've made too much. But over time, as you move to the left, these clones get more and more different because they're consuming things at different rates, they're producing things at different rates, and also the abundance of cells, the viability of them is also changing over time. So you can see on the far left, the principal component analysis can clearly see that all those clones are differentiated by their metabolic profile. We can actually go into the data a little bit further using multivariate analysis. This is an example of an S-plot. So now we've taken the same data you saw in the previous slide, but actually asked a slightly different question and sort of looking at the difference between day 0 and day 16. So the metabolites that are changing the most are going to be represented at the extreme ends of the S-plot. So in this case, the bottom left part of left quadrant shows a number of points highlighted in red circles, and those are the markers of interest at day 16. So these are the markers that have changed the most over time at day 16. And so if we go into those and look at the mass spectrometry data underneath those, we'll see a number of targeted ions, things that we actually looked for in the library, but we also might find some peaks of interest that are unknowns, and these are unknowns that have changed over the time course and give us clues as to what's happening in the clones and how we can use those measurements to better inform our process in the future. So hopefully I've convinced you that the solution here is more comprehensive than previous solutions out there. At the same time, we've tried to maintain ease of use through a guided end-to-end workflow. We're also combining powerful informatics with routine high-quality data. What this results in is ultimately better decision-making for everyone. If you want to discover more about this solution or the BioAccord, please visit us at Waters. Much of the information I've covered in this presentation is covered in our recent application notes and posters. Thank you for listening. As Letitia mentioned in the introduction, cell culture media analysis is only a small part of what the BioAccord is capable of. We've also put the BioAccord in the hands of one of our partners, Sartorius, looking at drug substance in their bioprocess workflows. I now have the pleasure of introducing Charles Petraska to discuss the power of using BioAccord for CQA monitoring from AMBER-15 in process control.
Thank you, Mark, for that deep dive into cell culture media analysis. And thank you, everyone, for joining and your interest in our collaboration. So I'll give a brief background on myself. My name is Charles Petraska. I'm an upstream engineer for Sartorius, located in the US. So we're actually in Bohemia, New York. That's on Long Island. So me and my team, we work in upstream cell culture. So we're a cell culture lab. We test upstream cell culture equipment and design. So that's the background for our role in this collaboration going forward. With that, I'll kind of proceed into the presentation. So just a quick agenda and overall look. So we'll go through some of the goals of the collaboration, some of the background data that might be relevant, setup of some of the experiments, the results, including process data and LC-MS results, and then wrap up with a conclusion. Okay, so I wanted to start by just reading the text that's in the yellow box there, because I think it encompasses the spirit of the collaboration pretty well. So by coupling Sartorius Ambr 15 and Waters BioAccord, development engineers and scientists can generate accurate and precise quality data with minimal additional work, low sample volume, and sub 24-hour turnaround times. So that's really, that's the dream, right? To just be able to get all this quality data as fast as you want. You can just grab it right offline if necessary. So that's what we're trying to work towards here. So our team goals, as I said, we're an upstream lab. So we have a few different Ambrs, a few different multiple scale systems, but with the Ambr high throughput multi-parallel reactor we have, we figured that would be the best way to test this collaboration. So what we're trying to figure out in the beginning is how best to really put this through its paces, how best to provide the customer with the data that they would need. So how we were going to do that was we decided we would generate a range of MAB product quality data from our CHO cell culture. So we ran Ambr 15 experiments. We used a range of conditions to try and generate a range of product quality data to really test out what we could do here. We wanted to reduce that data generation and retrieval time from the current, which is weeks in some cases, to below 24 hours. And we wanted to establish an ease of use for the end users and the workload on the team as a whole. Wanted to consider some of the differences between frozen and unfrozen samples. Normally, these samples are passed over to analytics, right? So a lot of times the throughput isn't there, or you may be transferring a lot of samples, so they're normally frozen. So we wanted to test out some of the differences there. We also did some initial trials bringing the data back into the Ambr. So this way the control software could actually see the LC-MS data, so some quality data back into the control software. We also wanted to try to then employ this data as a process trigger. So I found this to be probably the most interesting opportunity that's granted by doing this coupling here, is you can actually grab the process data and the quality data together and make changes to the process. So moving on, I'll just give a brief background of some of what CQAs are and some of the different development models that are in place today, and how we're seeking to change those to streamline everything. So why do we care about CQAs? What are CQAs? So CQAs are important because they can change a lot of aspects of the product. They can change immunogenicity, efficacy, required dosing, clearance rate. The FDA also has standards for certain biosimilars. They want biosimilars to attain similar attribute profiles as their branded counterpart. CQAs can also be a sign of cell culture health performance. So there's a link, a correlation between cell culture health performance for cell line selection and cell line development between CQAs and cell culture performance. So what are the potential benefits of coupling the Ambr 15 with the Waters BioAccord? So in upstream, analytics is our friend, right? Analytics generates all our key data. So with them, we wouldn't really be able to proceed. So anything to reduce load on the analytics team is extremely helpful. So what we are trying to do is generate otherwise infeasible data with this, which would be mid-batch time points. So it's difficult to go to analytics and say, hey, we have 48 reactors, we're going to sample them every day for 3 different experiments. Can you run CQA analysis on every single one of those samples? It's just not really possible. with the current motif of sampling. So what we're proposing is coupling these two systems together and using it as a complement to analytics. So not necessarily replacing analytics at all, actually kind of developing with them and using it as a supplement, something that we can use as a screening tool in the upstream lab to get kind of these rough sample measurements that we normally otherwise wouldn't be able to get. What this opens up the possibility for is pretty interesting. It opens up the possibility for some quality-driven feedback. So process parameters that would change depending on quality data. So you'll see later on in the presentation how powerful this can actually be. And without this coupling, it's pretty difficult to do. So with that, what I wanted to actually do is kind of review what the current system looks like, right? Like what we're trying to do here. So, and on the top, we have a figure where I'm outlining different cell culture runs, and this is a normal traditional development path for upstream cell culture. So we'll say a MAB product. So we have a cell culture run that lasts about 2 weeks. You can see on the x-axis here, so in the yellow box, we have our cell culture run. At the end of that, samples are sent over to analytics for quality analysis. Those samples then have to get analyzed, and depending on your particular group and your particular analytics team, this could take anywhere between a few days and a few weeks. So if you follow this pathway, in between that time when you send your samples out and when you receive that data, you're not going to just sit there. So there's usually time to do 1 or 2 culture runs, and you haven't gotten the data back from the last one yet. So normally assumptions are made or tests are done on different aspects of the project. And then you get your data back a few weeks later, and you can implement changes based on that data or use it to confirm something that you might have assumed. But that doesn't have to be the case. So when we couple these two instruments together, we're able to actually send out samples, generate that data, and get it back within 1 day. So that's the kind of new motif that I have going on right here in this lower figure. If you can see on the lower one, we have, let me grab a laser pointer. So we have our cell culture run, quality samples are sent out to yourself basically, and then they would the data would return right back. So this could allow you to implement changes immediately right after the run, or what I find even more interesting is implementing changes mid-run. So since this quality data is given back with this amount of haste, you can actually use that data to implement changes in the process. So if we go further, I'll overview how we tried to do that. So we kind of said to ourselves, how are we going to really test this out? What's the best way to do this? So as I said, we selected our Ambr 15 for its high throughput nature. So anybody who works in upstream with Ambr units will probably be used to the bed layout here on the right. So what I've included in this presentation is 2 studies that we've done. The layout you see on the right is from the first study. So the conditions that we have here include controls, of course. So you can see them inside the little reactors here on the deck layout. We also included a high glucose condition, so just increased glucose to see the impact that would have on the key critical quality attributes, which in this case was glycosylation. We also included some glycosylation modifying supplements, so positive controls basically. One of them was chifunincine, the other was what we dubbed SUP2, which was just a mixture of a couple of metabolites and minerals and things like that. One of the other things that we wanted to test, as I kind of alluded to earlier, was that feedback. So I've kind of have this little clip art down here on the bottom. You have your BioAccord on the right and your Ambr on the left. So you get your data back. It goes through the BioAccord, and then you can either process that data using multivariate analysis, or you can just implement feedback directly back into your process. So that's what we did with this study 1 that I have over here. So when we received our glycosylation data back from the BioAccord the next day, we actually varied the glucose in the feedback conditions if we saw a deviation in that glycosylation. So we looked at the control condition glycosylation, and if we saw a deviation between that and the feedback condition, which started out as high glucose, we then dropped that glucose down to the control condition to see if we can kind of rein that glycosylation pattern back in. We also included some forced feedback conditions. So what those were is they started at high glucose as well, and then on day 3, the glucose was reduced back down to the control condition regardless of glycosylation, just to kind of see what would happen in case we didn't see a change. In the second study that I've included in some of the later slides, it's a much simpler study. That's why the bed layout isn't here. It includes basically 12 reactors. So 6 of them are all controls, so an N of 6 controls, and then 6 temperature modification conditions. So we actually modulated the temperature in that study. In study 1, the temperature was constant in culture station 1. In culture station 2, the temperature was shifted down on day 3 to 33 Celsius. So that's the difference in color that you see here. So just that one solid shift on study 1, and in study 2, we actually modulated the temperature based on the glycosylation profile. So speaking to the sampling, because we often get questions on this, I've included the normal upstream workflow that we have up here. So Amber 15, right here, you take your Amber 15 metabolite samples, which is a normal upstream day. You take these samples, you perform a clarification, which in most cases is centrifugation and filtration, and then you pass those samples off to your metabolite analyzers. So in our case, the CDEX Bio. So that's what's normally done. In this case, we aliquoted off 50 microliters that then went into sample vials and into the BioAccord. So once they were in the BioAccord, they were run overnight, and then we were able to view that data the next day. So the additional workload that's here is about 15 minutes on a full-time employee. So it's pretty minimal, the amount of workload that was done. I want to kind of just bring up that we did not do protein A here. I know that's commonly used in analytics. So with this, it's just the clarification step, and then the sample is passed right into the BioAccord. So again, only 15 minutes additional time, extremely low sample volume. You know, from my personal experience, 50 microliters is a pretty small sample for cell culture. In analytics, that might be not considered a small sample, but for us it is, especially when you work on some of the larger scale reactors. But what this kind of shows is the ease of incorporation that the BioAccord has into the Amber workflow. The Amber workflow itself, it's just these few additional steps as of right now. So we'll pass into some of the data. So I want to give some of the process data first. This isn't LC-MS data. This is normal process data, just so you can get a feel for how the cultures were doing. So over here on the plots, I know they're kind of small, so what I'll do is I'll just kind of talk through them quickly. On the left, we have 2 plots. Both of these are from study 1. So the first plot on the left, you can see that our orange condition is control. So we had maximum cell growth performance. This plot is VCD, so viable cell density, so showing cell growth performance. We had maximum cell growth performance in our control condition, followed up by one of our supplement conditions. Then in the middle here, we have our feedback conditions, so where the glucose was high in the beginning and then dropped down mid-run, depending on glycosylation feedback. And then underneath that, we have high glucose. So this was just high glucose throughout the entire batch. And then on the bottom, this was another glycosylation modifying supplement. If we move from left to right, the plot in the middle, this is all the same conditions, all the same colors. The only difference is the temperature. So the temperature was shifted for this culture station to 33 Celsius, so shifted down on day 3. You can see an overall decrease in cell growth for that culture station, which is to be expected when the temperature is dropped like that. On the right, we actually have the final plot, which is the VCD plot for study 2. So again, this was an N of 6, so just 6 control reactors, and then 6 reactors in culture station 2 that had their temperature modulated depending on the glycosylation profile. So you can see that the temperature modulation was down in this case, so cooler reactors. So that did decrease cell growth performance in those cooler reactors as well. So again, a little bit more process data here. So this is titer. So overall titer in grams per liter. So throughout the batch days. So we have our orange condition actually in the middle here. So our control condition landed in the middle. It was actually outperformed by our feedback conditions. High glucose condition landed right around control, followed by some of the glycosylation modifying supplements. Again, for study 2, the center plot here was the temperature shift down. So we saw a general decrease in titer for that temperature shifted conditions, but overall the pattern kind of remains the same in the order of titer production. For study 2, we saw a increase in titer production for our temperature feedback condition. So this was actually a slightly different process than study 1. So we wanted to try a different process as well and see the effect. But overall, a slight outperformance in titer for the temperature shift conditions and right around the middle titer production for our control conditions. So now we'll get into the actual LC-MS data, so the glycoform data. So this data was generated using the intact mass assay on the BioAccord, which allows us to run it very quickly, get our data back very fast. What you can see here is mass chromatogram. So this is a typical antibody kind of shape that you see here. As I said, I'm an upstream engineer. I don't work with LC-MS very much, but after some basic training from the Waters team, I was able to kind of wrap my head around the fact that these peaks you see on the right, these are the glycoforms themselves. So the integral of these peaks is the relative amount of each glycoform. The black that you see on the plot on the right, that's actually the control condition. So these are the different glycoforms, the major glycoforms that we saw in the control condition. If you move even further to the right, you'll see some purple lines. That's the glycosylation modifying supplement, chifounicine. So we saw that there was a large shift in glycosylation profile for those supplement conditions, which was expected. That was a positive control. So that checked off that box. The BioAccord was able to detect that pretty well. So this was kind of the glycosylation profiles for all the conditions through all the days for the major glycoforms. So there's a lot on this plot. So I'll just kind of talk through what everything is here. So if you go through the columns from left to right, you'll see we have our control condition. The next column is one of our feedback conditions, so high glucose in the beginning and then reduced glucose when we saw a shift in glycosylation profile, followed by force feedback, high glucose throughout the run, and then supplement 2, which was a glycosylation modifying supplement. So if we go throughout the rows, these are the different major glycoforms, and the y-axis here is relative percent abundance. So out of the amount of MAB that was produced for the middle of this plot here, let's say this time point, we'll call it day 3 for the control condition on this top left plot. You can see that was about 20% relative abundance G0F, G0F. So that's how to kind of make sense of what's going on this page. But then if you kind of follow this across, you can see some really interesting things happening here that are kind of difficult to see when you're only sending out end of batch time points, which is what I'm used to doing upstream work. So if we go from left to right, on this top row, this will be G0F, G0F relative abundance throughout the conditions. So if you look at the orange line, that's the control condition, the control culture station. So that's where the temperature was left at our control temperature throughout the batch. If you go from left to right, you can see a general rise in this G0F, G0F concentration between the control condition and the feedback conditions, and then even a little bit steeper of a rise towards the end for the high glucose condition. Supplement 2 had a greatly decreased G0F, G0F percent relative abundance, and you can kind of follow these along as you go down the different rows and see the effect that these glucose conditions had. What's really interesting, though, that we didn't realize was going to have such a strong impact was this temp shift. So again, this is that study one where the first culture station stayed at control temperature, and then the second culture station was temperature shifted down to 33 Celsius on day 3. So those conditions, those temperature dropped conditions, those are in blue. So you can see right after about day 3, a large deviation occur in most of the major glycoforms here, which was really interesting that we could see that difference so quickly, and that it carried through throughout the different conditions and throughout the batch. So this is just really great data to have, and it's difficult to kind of get a feel for how your process is going to do depending on different conditions without plotting these throughout time, which is relatively difficult when you're sending out samples to analytics and it's this quantity of samples. So you can kind of extrapolate from here what this can be used for if you were targeting a specific glycoform profile, specific glycoprofile, if you were making a biosimilar, or you can even extrapolate this to other key quality attributes that you can kind of plot across time. So on this slide, we'll go through study 2. So this was a little bit simpler, but in my mind, it was just as interesting. So the first plot that you'll see on the left, this is the same as the top left plot on the last slide. So just to get a zoomed in view of that. So the gray section here, those are the days that the temperature was shifted down for the blue condition. So you can see a few days after the temperature was shifted down, we see this large deviation in glycosylation profile. So the G0F, G0F relative abundance goes up for that condition. And then knowing that from our first study, we tried to learn from that, and we said, can we target an exact G0F, G0F relative abundance in the product, in the main product? So we picked a target. We tried to pick something in between our control and our temperature shifted control, which was about 30% is where we landed on to try and target. So what we did is we dropped the temperature in that blue line there. Again, the orange is control. So if we're on the graph on the right, we dropped the temperature to 35. If you can see this thin gray line right at the start of the plot. So not as strong as the temperature shift as the study before, because we wanted to kind of end up right in the middle of there. And then each one of these gray sections that you see, that was another temperature modulation. And you can see at the bottom what that temperature modulation was to. So again, the entire goal was to try and target that 30%. We got pretty close towards the end, and then we actually overshot. So we brought the temperature back up to try and bring that relative percent back down. Didn't quite make it, ended up landing at 32%, which initially we kind of took as a loss, but in reality, we actually ended up right in between those 2 points. So within one study and one iteration, we were able to get right next to that target, which again, you can extrapolate the use of that for your own work. But that ability is pretty powerful when you couple these two instruments together. So this I found very interesting. So through our work with Waters, we actually discovered, again, we're upstream engineers, so we're not familiar with all these analytics assays. So normally protein A is done, protein A purification is done on these samples. In this case, it was not. So what that allows us to do is get the data back very quickly, but it also allows us to see things that are in solution that otherwise would be missed. So this is a total ion chromatogram with retention time on the x-axis. So you can see there's two major peaks here. The first one was identified as the light chain, so just free light chain floating around in solution. And the second one was identified as the MAB peak itself. So again, you wouldn't really be able to see this light chain if protein A purification was done. Why is this relevant? So you'll get to see in this slide right here. So in analyzing this light chain itself, we found that there was actually modifications to it. So it wasn't just one pure conformation of light chain. So I kind of have these little pictures down here on the left to kind of illustrate this. So the one on the left, this is just a pure light chain, and these are some of the modifications. So you have circle, diamond, triangle, and then just one that has some pieces clipped off. So again, this graph is laid out in the same manner. So we have the different conditions as the columns, And then instead of having glycosylation relative percent, this is light chain modification relative percent. So we saw a different modification for each condition. So if you follow, let's say again, the control condition, so the orange from left to right, this is the light chain plus 134 Daltons. You can see that there's a difference in the amount of that light chain modification as you go through the conditions. So the conditions were actually correlated to these light chain modifications and actually much stronger than the correlation than the glycosylation to the conditions. So if you look at the second row, this is the light chain plus 305 Daltons. So you can see the control condition, it ended up spiking up around day 8 at about 60% relative percent of the light chain was in this plus 305 Dalton conformation, while the temperature shifted condition was down around 20%. So that's a very large deviation there, and you can see this correlation kind of plays through all the conditions. Again, just one of the benefits of doing it this way and getting this extra information that's otherwise kind of just washed away with purification. So getting near the end, I'll just kind of review quickly some of the other things that we did. This is the impact of freeze thaw. We didn't really see much of a difference. So since we aren't freezing these samples, we wanted to just see if there is a difference between freezing and not freezing. It looked like we didn't really see much of a difference in some of these lower level glycoforms. So the red is the data that you've seen throughout the presentation. These samples were never frozen. The green and blue, these samples were frozen and filtered versus frozen and not filtered, respectively. So if you look at them, you can see a slight difference in some of the higher order glycoforms. This may be process or product specific. Further investigation is needed, but it's just information I thought might be important to include in the presentation as well. So onto just some conclusion statements. Coupling these two units together, the AMBER 15 and the BioAccord, we were able to see deviations in these CQA, specifically glycoforms, as early as day 4 using our AMBER 15 process. We were able to see that there was light chain modifications that were strongly correlated to process conditions. The impact of freeze thaw was minimal but still visible for some glycoforms, and the close coupled LCMS feedback could be used to direct glycosylation profiles. So we successfully directed the glycosylation profile using just one round of testing and one iteration to target a specific glycoprofile using our AMBER 15 process. The full turnaround time for 24 vessels, we often get a question on this. It's under 18 hours actually, methods being advanced kind of every week. We have it around 10 hours now, so you can make subtle tweaks to that method and kind of reduce this time even further depending on your workflow. And that kind of brings us into how much workload is on the team. The additional workload for this coupling is around 15 to 30 minutes, depending on who's doing it, how many samples there are, but it's pretty minimal. So the impact on the team is pretty minimal when adding this analyzer onto that. Some of our future work, we'd like to look into some of the other CQAs, so fragmentation, aggregation, charge variance, some things like that. We also want to pull this data back into our SIMCA software to do some multivariate data analysis on it to try and model what's going on so we can really harness some more of this power. We also want to employ similar testing on a range of additional biotherapeutics. We're actually bringing in some more cell lines to do some additional testing. So if you want to find some more information on this for AMBER sales, you can contact our Sartorius account at bps-requests@sartorius.com. And with that, I just want to thank everybody that was involved in the process, and thank you all for attending today, and thank you all for your attention. I think Mark and I will take some questions now.
Well, thank you very much, Charles, and thank you also, Mark, for these two very insightful presentations. So now we're going to go to questions. We have a few questions to answer here, but if you have additional questions, please do not hesitate to type them in the Q&A on the screen. So the first few questions were more about the sample preparation, and maybe starting with Charles. We would like to know why you did not use protein A purification And if you think that if the samples were to be purified, you would see any differences in the data.
Thanks, Letitia. So we actually did test the samples through protein A purification at Waters as well to just see comparability, and the samples were comparable. The reason why we removed the protein A purification step was kind of to streamline that sampling process. This way we can get that data back as fast as possible, and there really wasn't much extra work for the full-time employees in the lab.
Okay, great. So staying on sample preparation, maybe this time to Mark. Could you tell us a little bit more about how the spent media samples were prepared and if you used any kind of derivatization?
No, the methodology is pretty simple, Letitia. So we employ direct analysis. Didn't use derivatization here. Waters does sell some excellent derivatization kits and products and solutions as well. But for the BioAccord solution I described here, we wanted to see how far we could push the technology for direct and simple measurement. In doing so, in this case, we can capture a much larger amount of information about the other components in the mixture, what they're doing over time relative to the main feed components. And then finally, the sample preparation itself is generally very straightforward. We typically take filtered samples, straightforward dilution. Generally, the BioAccord is pretty sensitive, so we dilute 50-fold, 100-fold typically. I think it was 100-fold for most of the data you've seen today. And then we run the samples that way. So.
Okay, great. So both of your methods seem quite simple to implement. So we were wondering if you would need to re-optimize them every time you change product or cell line. So I think the question is for both of you. Maybe Charles, if you want to start.
Sure. So overall, it is a pretty simple method. Preparation, everything for the project overall is pretty easy. As far as kind of converting that and moving it over to a new cell line or a new product, you wouldn't need to change too much. So the overall method, most of the components in it, for the intact mass assay specifically, the only thing that would really need to get changed would be some of the sequence information for your actual product, but overall most of it could stay the same, so you really wouldn't have to make too many changes there. So for that specific assay, and I believe the same is true for the media analysis as well, but Mark could probably elaborate further on that.
Yeah, so for all the work we've done so far with various collaborators, we use the same method. So we have a positive ion mode method, which is mostly what we run. We also have a negative ion mode method, but it's very similar. We run them again and again. It's not much change. We want to keep consistent methods for measuring cell culture media analytes so that we can compare them over time. So again, because we dilute the samples so much, that's quite an advantage for LCMS. We don't have to worry about changing the methods too much. So for the compounds you've seen here listed, we run the same methods over and over again.
Thank you both. So Charles, you showed us how powerful the BioAccord system was in conjunction of the AMBER system. Are there any other attributes you could measure on this system?
Yes, so the BioAccord is a full LCMS system, so it can measure a lot of different attributes, aggregation, sequence variance, anything that you would use an LC for, you could also use the BioAccord for. So there's a few different attributes there that can be measured. We selected to use the intact mass assay for our studies because it gives us the glycoform data back pretty quickly. There's minimal sample prep there. But you can also do the cell culture media analysis that Mark was describing. They use very similar mobile phases, so the switch over from the intact mass assay over to the cell culture media analysis would be pretty easy as well. So a few different options there for you, a few different configurations. We're actually moving into that phase of the collaboration. So we're actually beginning to test the cell culture media assay that Mark has described over in our labs. So we're actually going to start work on that. As I said, it should be a pretty seamless transition though.
Great, thank you very much. So Charles, if I remember well, you weren't an expert LC-MS user prior to getting the BioAccord. So can you tell us a little bit more on how easy the system is to use and if it can function as a walk-up analyzer?
Gotcha. Yeah, for sure. I was definitely not an expert LC-MS user. I had never used an LC-MS before, so I think that kind of speaks to how easy it was to use. Overall, the setup for the instrument took about a week. During that time, the Waters employees that were setting it up, they trained us on the system itself. So me and our team of 6 people, we were all trained on it within that week, and everybody was able to operate the system directly after that. So the BioAccord itself is a very easy system to use. It takes about 15 minutes additional time for our workflow, so not very time consuming and pretty easy to train people on as well. Yep.
Yeah, and I'll just maybe add to that as well. I mean, you know, Waters has a long history of making LC-MS platforms, and the BioAccord really is a culmination of all those efforts. And we strive to do everything that Charles has just described, you know, make it easy to use, everything from simple, straightforward calibration, much easier than we've ever done in the past, easy to do the basic maintenance on it and troubleshooting. So yeah, it's great to hear people like Charles and his group kind of, you know, succeeding using the BioAccord, because that's exactly what we designed it to do.
Well, thank you very much both for your answer. Maybe a last question about the future. So what do you think companies like Sartorius and Waters can do to continue to innovate in analytics in this bioprocessing space?
Yeah, so I'll start, Letitia. I mean, Waters, I mean, we're committed to providing streamlined technical solutions and making them easy to use, such as products like the BioAccord, while at the same time leveraging all the amazing information that LC-MS provides, and using informatics to take this information, distill it down to what matters, and help drive decisions. I think the opportunity for us to continue tailoring this product and other products around monitoring specific or broad metabolic pathways with respect to cell culture media analysis is an exciting future, and you know, we hope to continue to meet the demands of all of our bioprocessing customers and collaborators.
Yeah, just to follow up to what Mark said, from the Sartorius side, I believe we try and really get a feel on what the customers need and what would help them the most. That's one of our real key traits there. So I think this is a perfect example of this collaboration itself. So our collaboration with Waters really tries to address a customer need that we see, kind of ease that pain point for the customer, getting this data back quickly and having this entire system very easy to use. So more collaborations like this would be kind of the thing that we would do.
Great, thank you very much. Looking really good for the future. So thank you to both of you. Thank you all for listening to this webinar. If you have further questions, please do not hesitate to contact us, and we'll get back to you. Thank you. Bye.